to something that at one point they might have been resistant to. So we turned to a drug which is actually well known in the diabetes field, which is called metformin. So metformin reduces insulin levels, modifies cellular metabolism. One of its targets is AMPK, which is activated when you exercise and you kind of use up all your nutrients. But it turns out it also inhibits the electron transport chain. And in fact, some studies of mice have shown for cancer that if you use metformin, you can actually inhibit tumors, and it can be reversed by putting in a yeast complex 1 subunit, which is insensitive to metformin. So we know that at least one of the major targets then is electron transport chain complex 1. And I just told you that BOC1 actually regulates electron transport chain. And if I take BOC1 down, I'm inducing complex 1. What's interesting is the therapeutic efficacy of metformin is not quite clear. There are actually a lot of clinical trials going on now, but I would suggest that we have no idea which patients we should be selecting. So the results have been mixed, although they're getting more promising. So we said, okay, what happens if we do this? Take down BOC1 and then try metformin. So here's a case where we're trying metformin and we haven't taken down BOC1, and basically for tumors, this is tumor volume, hasn't done a thing. But now, if we take down BOC1, then actually with metformin, we can reduce the tumor significantly. So this gives you an idea of the shrinkage in size. And this gives you an idea of a graph over time, where this is where we've taken down BOC1 first and then treated with metformin. And this is a case where we haven't touched BOC1 and metformin really doesn't do anything. So that would look pretty good. But, you know, the problem was we took down BOC1 with shRNA. People somehow don't get shRNA the way we treat mice. Actually, we would be done, by the way, if we could experiment on people the way we experiment on mice, but we don't. So we kind of had to say, okay, can we do this another way? Is there some kind of therapeutic way that we can take down BOC1? And amazingly, there was. So it turns out there's a drug called panhematin. It's actually an FDA-approved drug, and it targets BOC1. So just to tell you about panhematin, it's a lyophilized form of an alkaline heme for a disease called porphyria. And it corrects the heme deficiency in liver and represses production of porphyrin precursors. Why does that work? Well, I told you that BOC1 was actually known for regulating redox, and this is part of the reason. Here's BOC1. It actually inhibits an enzyme called heme oxygenase. Heme oxygenase breaks down heme. Heme, when heme is high, can bind to BOC1 when BOC1 is sitting on the promoter, cause it to be exported out of the nucleus, whereupon it gets ubiquitinated and degraded. So this is a very nice little negative feedback loop. If I overload the cell with heme, I can break down BOC1. And here's an illustration where we did that. We added heme, and you can see on the western that BOC1 goes down. This is heme oxygenase, mRNA. So if we add heme, and BOC1 goes down, remember it was suppressing heme oxygenase, then heme oxygenase goes up. So it works in our cells. I said, okay, let's try this. So can we exploit now heme treatment of BOC1 as a new type of therapy? So we tried this in 2 types of models. One was a so-called xenograft model, where you take a mouse. The mouse is a nude mouse because it has an immunocompromised system. If you didn't do that, then the tumor would be rejected if you put a human tumor into a mouse. So this allows you to put a human tumor. In this case, we used a cell line called MDA436. And as you can see, if you look at the normal growth of the tumor, doesn't really change if you put heme in, doesn't really change that much. Actually, it changes a little bit if you put heme in, doesn't change that much if you put metformin in. But you actually get a dramatic change when you put them both together. And in fact, when we've analyzed these types of mice, we can see that some of them are completely cured. We don't want to say that this is the ultimate cure because, of course, some of them have a bit of a bypass too, but you're definitely doing much better. But right now, the standard is, okay, that's a cell line, you know, it's been grown in culture, not so good. What about patients? So So we have a new tumor model called a PDX, patient-derived tumor xenografts. And that involves basically a hunk of patient tumors that have been put into mice and never put on a culture dish. They're just transported from mouse to mouse to mouse. So essentially, for a patient, it's their mouse avatar. And so we asked, okay, what about that? Now, these are real tumor cells. What happens there? So here's an example. And this is one where we knew that BOC1 was expressed in the tumor. And so again, metformin doesn't really do anything. Heme does something. Heme does have some other targets, although we have a heme-resistant BOC1, and we could show the effect we're looking at. It seems to be fairly specific. And then again, if you put them together, you get a really dramatic decrease. So just to summarize what I've told you so far, I showed you before, this is the basic component of metabolism. Again, compatibility between PC and MAC. Nice to know that it hasn't totally changed, that they're incompatible. This is supposed to say metformin. So this is the basic scheme I told you. I haven't really shown you, but we know that BOC1 can target also some aspects of glycolysis. We've actually done so-called seahorse experiments, looked at respiration, looked at lactate production, and we know that at the same time that we're increasing oxidative phosphorylation, we're also decreasing production of lactate. So we really are shifting, kind of like seesaw, the balance of the energy pathway. At the same time, we also know, and I really didn't show you, is that we're affecting this step that leads you to acetyl-CoA. See, I can't get away from acetyl-CoA. And BOC1 actually inhibits the ability of pyruvate dehydrogenase to work, so when you take it down, now you're essentially moving pyruvate down this pathway as opposed to lactate. And we also know that BOC1 is even affecting aspects of the TCA cycle, tricarboxylic acid cycle. So, but what I showed you is if we inhibit BOC1 with heme, then we can make the system sensitive to metformin. So that's very well, and that works for breast cancer, and that works in preclinical mouse models. But what about people? Again, we have to kind of go back to people. So can we use BOC1 to first identify patients who benefit from metabolic inhibitors and then potentially try it? And so what we did was go to a new database that's now available to everybody called the TCGA, and there's a few others as well that we've looked at. And it's a great resource because you can get thousands of patients, or a couple thousand patients, and you can look at their gene expression patterns. And then you can actually organize them. You can ask questions about relationships. And I'm going to skip a few things, but what I want to show you is actually an individual patient level. So here's a scheme where you can now plot different genes, like expression, and BOC1 is the top. And going vertically is individual patients. So you can actually ask what's happening with each individual patient. So pink is high expression, blue is low expression, and gray is somewhat in between. And all of these genes down here are different genes within the electron transport chain. So I don't actually have to tell you anything for you to come and say, boy, I think I get this gene signature. When BOC1 is high, electron transport chain genes are low. Now imagine you're using metformin and you don't have any genes that are expressed to target. It's not going to work, right? So if you're actually taking this whole population, here's a whole population we would predict are completely resistant to metformin treatment. But we think if you actually use heme first, then in fact it would be sensitive. This group here is BOC1 low. And as you can see, I mean, depending on the patient, you get different levels of expression, but overall high of electron transport genes. And we would say, these are the guys that you want to target with metformin. Go for those guys. And these guys are somewhere in between. Maybe we can make them better with heme. Maybe we can find another scheme that takes advantage of the same ideas that we were doing before. So we said, wow, that's great for breast cancer. What about other cancers? I'm not going to show you the pictures, but we can see pictures like this for a number of other cancers. Here's some examples. What we did here was just say something simple. If we look at all the genes in different cancers, and we start with BOC1, and just say, what genes are most inversely correlated in expression with BOC1? So here you would say, oh, it's all of these guys, because BOC1's high and these guys are low. So we did that, and we asked that for colorectal cancer, for liver cancer, for lung, for skin, ovarian, pancreatic, prostate, and breast, all or breast TNBC. And here we just said, okay, if you take those genes now and ask what do they correspond to, what are they? The majority fall in the category of oxidative phosphorylation. And in fact, the majority are actually genes in the electron transport chain. So we know that what we're looking at for breast cancer really has application more broadly. And again, we're not getting all patients, but it allows us to stratify them, and we think to actually come up with a new strategy for the most resistant ones. So to summarize, this one actually worked. I don't know why. If we start out with a heterogeneous tumor, then we can actually get rid of one that expresses BOC1. So our BOC1 levels are high. We can reduce BOC1 levels using SI or SH BOC1 or heme or potentially targeted therapeutics. We can get to a more homogeneous tumor state, and that now makes it sensitive to metformin, and we've also done some other treatments. And so we can, some cases cure, some cases get to a much better stage where we're taking advantage now of oxidative phosphorylation, and potentially we can also take advantage of ROS. So we would suggest sort of this strategy for cancer of kind of a two-part, let's go from metastatic, highly diverse and so on, to non-metastatic, more homogeneous, and then let's hit it with more novel therapies like even chemotherapy or radiation. So I want to end with Gobin. Gobin wrote a book, many of you have, which is his selected papers. He worked very hard on that, and he actually summarized each collection of his work. And if you haven't seen this, I would suggest that you do. What struck me in part was kind of the preface. Okay, who did he quote? Louis, and he said, "We must be modest, except in our aims." And I think that really is Gobin in a nutshell. So with that, I'd like to acknowledge the people in my lab who did the work, some very talented postdocs. Casey Frankenberger started it. Jiang Li really did most of the metabolism work I talked about. And Anjin Li was this great undergraduate, physics undergraduate, who was flunking out. And I asked him to come to my lab and to start working on some of these computations, and he did all of our gene signatures. He's now a star graduate student at the University of Chicago. So thank you, Gobin, and all of my colleagues from the Corona days. Thank you very much. I don't know, probably we don't do questions, right? Keep going.
You want to answer questions?